Hello and welcome. My name is Alberto Williams, and I'm a master's graduate from the Department of Physiology and Biophysics of Case Western Reserve University. Under the mentorship of Dr. Damir Llanero, I will be guiding our discussion of the electrophysiological properties of the mammalian central nervous system. We hope that this presentation can complement your reading and learning experience as we venture into the electrophysiology of the brain. Let's begin. The neurosurgeon of today has at their disposal several different techniques to explore the physiology of the human central nervous system. This slide illustrates the spatial temporal span of commonly employed techniques. Starting from left to right, we find the patch clamp technique, which consists of many variations to record activity from individual synapses, dendrites, or neurons, with a millisecond time resolution. However, this technique would not be useful to study lengthy processes in the hour-to-day time domain. Stereo EEG, or SEEG, is an invasive technique with a broad temporal limit, but somehow limited temporal performance. From the imaging standpoint, fMRI is useful for a large number of time-dependent events occurring at the whole brain column level. Despite this, fMRI is not useful for fast-occurring neuronal and synaptic events due to its poor temporal resolution. On the other hand, non-invasive comparable techniques like EEG and MEG have similar time and size intervals, but lack well-defined spatial resolution. We will also further discuss the differences between invasive and non-invasive techniques further in our discussion. Whether it be for research or diagnostic purposes, obtaining information about brain activity is of utmost importance, and to keep innovating and making our current techniques more efficient will be considered a milestone in basic neuroscience, neurology, and neurosurgery. For the purposes of this chapter, we refer to NPI as any qualitative and/or quantitative method that provides a measure of brain activity, both regional and at the cell level, that delivers insight about electrophysiological phenomena in any illustrative form. In short, we refer to NPI as any method that can provide both numerical and observational data about the activity of the brain in order to draw research or clinical conclusions. We recognize that many techniques we name under NPI also fall under the classification of neuroimaging. Notwithstanding, we can all agree that accessing the brain is quite a feat, yet the data we can obtain is highly valuable. With this said, let's proceed to differentiate invasive and non-invasive forms of NPI. It is common use in the medical field to use the word invasive to indicate surgery or surgical involvement, meaning that there is a necessity to inwardly access the body of the patient. On the left side, we should note that invasive methods may pose the highest risk towards the patient because we're directly contacting the brain region of interest, most commonly by inserting a probe or a microelectrode. Nonetheless, this is the prime option to obtain the most accurate measurement of neuroelectric behavior. On the right side, lower risk methodologies in neurophysiology, i.e. non-invasive, involve indirect measurements of neuronal firing. This can be done, for example, by attaching scalp electrodes and comparing the signals to a reference electrode. Given that non-invasive methods are widely used for research and clinical diagnoses, the chapter will mainly highlight the discussion of these techniques. Regardless of how brain activity is recorded, we know that these activities in neurons and glia alike cause changes in resting membrane potential. These changes are due to several factors, including voltage-dependent ion channels, agonist or ligand-gated ion channels, intracellular event-gated ion channels, ionic permeabilities, among other types. One of the main mechanisms that allow for the repeated activation of these events is the restoration of steady-state values. In other words, for every electrical event done by a neuron, there is a mechanism to restore the original or resting conditions. The main mechanism involved in this is a sodium-potassium ATPase, which consumes energy to counterbalance ionic changes occurring during the action potential or other activities in the brain. The exorbitant amount of energy required by mechanisms like this pump is the basis of fMRI and neurovascular coupling. An early electrophysiological technique consisted of penetrating cells with sharp glass electrodes connected to an amplifier to measure electrical activity in a given cell. The figure illustrates two examples in C, namely a voltage-activated and voltage-inactivated ion channel, and the channel activated in the presence of an agonist. Two fundamental variants of this technique can be used. One is referred to as voltage clamp and consists of forcing a certain voltage across the cell membrane to measure the currents evoked in the process. The second being a current clamp, which instead uses a protocol to impose a current value across the same cell membrane to measure the resulting voltage change. Both were equally instrumental for the discovery of the basic functioning of mammalian neurons and glia. Modern electrophysiological techniques can be paired with high-definition MRI to record responses of neurons located in the proximity of multi-contact electrodes, commonly used for deep brain stimulation, or DBS. We can notice that this approach allows to morphologically and electrically define the presence of certain clusters located in different parts of the brain. This allows for the characterization of pathological responses in various centers with usually normal morphological appearance. Electrophysiological data can be presented by a variety of methods. Parts A, B, and D depict frequency domain plots, where events of a certain amplitude, indicated by the color scale, are plotted against the frequency at which they occur, and the time of occurrence during the recording. An example showing the usefulness of this technique is seizure detection, shown in B, or detection of singles firing in the subthalamic nucleus, shown in D. These plots were superimposed for clarity, and to show the actual voltage recording from which the time domain particles derived. The figure shown on this slide depicts the process underlying deep brain stimulation, or DBS. DBS is a neurosurgical procedure by which an electrode is implanted into a region of the brain to aid the patient in attaining neuronal control. In this case, we observe the subthalamic nucleus on part C of the figure, shown in green, and this is done to improve control of movement in patients with Parkinson's disease. This is an invasive measure of modifying brain electrophysiology. The insertion of the electrode can be achieved by using computational modeling and MRI scanning, such to execute an insertion that will not rupture vessels nor damage cortical and subcortical regions. Let's start off our list of non-invasive MPI techniques by comparing EEG and MEG. EEG is considered the gold standard for studying neurophysiology, widely used to monitor sleep pathologies and epilepsy. EEG works out by attaching scalp electrodes and recording electrical potentials evoked by neuronal firing. This renders out as a spectra, which shows multiple waves, i.e. brain waves, which can be used to study the stages of sleep, levels of wakefulness, attention, and neuronal synchrony, or desynchrony. On the flip side, we have a newer contender called MEG. As opposed to EEG, it utilizes a headpiece containing a magnetometer. The electrical activity of individual neurons can evoke magnetic fields, which will be further discussed in the next slide. These magnetic fields can be measured by the headpiece by exploiting Faraday's law, which states that the flow of current generates a magnetic field perpendicular to its source. The direction of the magnetic field can be determined by using the right-hand rule. These fundamental differences yield a scalar value for EEG and a vectorial value for MEG. Given that vectorial values are composed of magnitude and direction, we can theoretically extrapolate the source of the magnetic signal via computational analysis. However, this does not significantly improve spatial resolution when compared to EEG. It is another set of separate analyses that have to be done. Another key difference worth mentioning is that the magnitude of an electrical signal recorded from the scalp is heavily reliant on brain extracellular fluid volume and resistance. This is why in patients who take diuretics, when we measure EEG signals, these can be accentuated, and the converse being true for patients with cerebral edema, where the signal can be nullified. Because the magnetic signals are not dependent on the volume nor resistance of the medium, we can be confident about the magnitude of the signal being recorded. Despite all this discussion, both techniques can be combined with one another and utilized in concert with fMRI. Now that we know the key differences between EEG and MEG, let's dive deeper onto how MEG works. On part A of the slide, we can observe a multipolar neuron conducting current. And we all know that current with respect to neurons is elicited by action potentials. And given that Faraday's law states that the flow of current induces a magnetic field perpendicular to its source, this means that each neuron's individual magnetic field is brought by action potential firing. On part B of the slide, we can observe multiple neurons firing, meaning that the magnitude of their individual magnetic fields can summate. Albeit intuitive, it is a summation of postsynaptic potentials, or PSPs, that actually contribute the most to these neuronal magnetic fields, and thus are the major providers of signals. Per computer modeling, a minimum of 10 to 50,000 cells are needed to evoke a detectable signal for MEG. In recent years, the number of neurophysiological studies employing the use of composite EEG fMRI modalities has been on the rise. It is quite exciting to see the level of innovation and ingenuity of research in order to address fundamental and pressing questions in neuroscience. There are at least two major forms of EEG fMRI composite methodologies: fMRI-informed EEG and EEG-informed fMRI. Starting with the former, as presented in the slide, we compile fMRI and EEG data separately, although both modalities can be done simultaneously on the patient, and this information is later rendered together. How so? Utilizing the superb spatial resolution of fMRI, we can determine the site of activity. But because fMRI is recording the delayed onset of neuronal input by measuring changes in oxygenation, we can inform ourselves of when the neuronal trigger happened by analyzing the EEG spectra. In other words, we use the data from fMRI to know where it happened, to guide our analysis of EEG data to determine when it happened. Therefore, we can accommodate for the resolution limitations of both techniques to acquire the whole picture. As we discussed previously, fMRI-informed EEG utilizes information obtained from fMRI to guide the analysis of EEG. However, EEG-informed fMRI uses a backwards approach, meaning that EEG data is used to guide findings on fMRI. fMRI events must be mathematically inferred upon first analyzing EEG points of interest. By using matrices and other linear algebra methods, the time course of fMRI events can be predicted and statistically correlated with EEG spectra. In a nutshell, the EEG data is used to inform us to look for where the event happened in the fMRI rendering. Let's now talk about a previous study which has revolutionized the field of neuroscience and has paved the way for new knowledge to come. In 1992, Di Pellegrino and colleagues made an interesting discovery in macaque monkeys. By using single and multi-unit recording methods via implanted microelectrodes in the inferior premotor cortex, they found that a group of neurons selectively fired during goal-directed hand movements. After more experimentation, they noticed that the same group of neurons became activated when observing an experimenter perform movement. These were later called mirror neurons, coined to describe these cells that fired in response to visual input of movement and in anticipation of. More studies followed confirming an extensive network of mirror neurons in humans, referred to as the mirror neuron network. The mirror neuron network is vital for achieving both cognitive function and movement mimicry. As you can tell from the slide, both neuron number 1 and neuron number 2 respond to grasping movement executed by the monkey. When the human subject performs the same action, we can see both neurons firing, and in the same manner with the film. You should also be able to notice that neuron number 2 seems to respond the most when compared to neuron number 1 when the grasping action is both performed by the monkey and after visualizing the human and the film, indicating the possibility that this mirror neuron network may be broadly tuned, much like ocular dominance columns in the striate visual cortex. Having explained the concept behind the mirror neurons, how has this knowledge brought new avenues for the advancement of neuroscience? A new subfield called Brain-Computer Interface Systems Engineering, or BCI, has emerged to find ways in which we can exploit the use of mirror neurons to bring back function to disabled patients. The slide shows cortical activation maps of a subject performing foot motor imagery, fMI, and foot motor execution, fME. The figure shows that areas of activation when observing foot movement correlate to areas of foot movement execution. Thus, this experiment provides the electrophysiological basis for designing BCI systems to aid, for example, paralyzed patients. How does BCI work? Let's suppose we have a patient who has undergone a T6 ablation, meaning that any motor control below the spinal cord level is lost. Despite this person not being able to walk, this person can still mentally visualize the act of walking. If this patient is connected via a headpiece to a computer, as we discussed before, the mere thinking of walking is going to activate the neuron circuits involved with the act, and this neuronal activity can be recorded via the computer interface. Using an exoskeleton containing an EEG that sends data to a processing software that can convert neural activity into executable robotic commands, it is possible to allow the patient to say, dorsiflex the foot. To continue investigating in this topic would provide groundbreaking treatments for disabled patients and revolutionize the field of neurorehabilitation. This concludes our chapter on electrophysiological properties of the mammalian central nervous system. We hope that this presentation has complemented your comprehension of the topics covered, and we also hope that it sparks creativity for ongoing and new research on the ever-evolving field of brain electrophysiology. Thank you.